On this slide, you're looking at what's called a karyotype. Karyotypes are simply the chromosomes that have been removed from a cell and placed in pairs in order to be examined. So obviously, we're looking at the DNA here, the chromosomes from a eukaryotic diploid organism. We know that because there are multiple linear chromosomes, and we can see that there are 23 pair. So this is DNA chromosomes that have been removed from a human cell. Humans have 23 different types of chromosomes. Each one of those types of chromosomes is paired. There are 2 copies. One copy was inherited from the mother and one copy from the father. The pair of chromosomes in 22 of the types are identical to each other. The 23rd pair, however, is a little different. The 23rd pair are the sex chromosomes. Female humans inherit 2 copies of the X chromosome. Male humans inherit one X and one Y. So this is a karyotype from a human male. Now, the other term to know is autosome. Autosomes are simply all of the different types of chromosomes except for the sex chromosomes. Autosomes contain all of the genes that code for all of the proteins that build the human body, except for the sex chromosomes. Remember, DNA performs 2 very different functions in a cell. First, it's used to make more DNA. That's the process we call replication or synthesis. And second, it's used for a process called transcription, which produces RNA. In DNA replication, the DNA molecule is replicated or copied along its entire length. Whenever a cell prepares to divide, it has to undergo DNA replication because each of the new daughter cells is going to need her own complete DNA molecule. Remember, we said DNA replication is semi-conservative. The DNA molecule first splits into 2 individual strands, and each strand is used as a template to make new DNA upon. Transcription is a very different process. During transcription, the DNA is used to make RNA. The DNA molecule will be used to make an equivalent but complementary RNA. Remember, only one strand of the DNA molecule will be transcribed to make RNA, and that's the template strand. The other strand is known as the coding strand. Transcription occurs only when a cell has a specific need for a particular protein. So we said inside the human genome, inside all of those chromosomes, there are approximately 3 billion base pairs. A lot of that DNA is genes. We estimate about 30,000 genes are in there. But the vast majority of the DNA in a human cell will never be transcribed. Remember evolution. The genome that is inside of your cells was inherited from previously existing cells. So throughout the time that life has existed on this planet, cells have divided and passed DNA on to the next generation of cells. Along the way, that DNA molecule is altered. So the DNA that's in your cells has been passed down through many, many, many millennia. And along the way, it has been altered. It's been altered with mutations, and it's been altered by the insertion of viral DNA. A lot of the DNA that you have in your cells will never be transcribed because it is no longer useful to the cells. They do not have a specific need for that specific protein. And some of it isn't even human. Some of it is viral. Our DNA is littered with DNA from ancient ancestral organisms. We no longer express it. It sits in the DNA molecule, but it is not actively used to build protein. On this slide, we're looking at the genetic code table. There are lots and lots of versions of this table available in textbooks and online. Remember, what this table shows us is the code in our DNA. Remember, DNA is used to make RNA through transcription, and RNA is then translated to make protein. And in translation, groups of 3 bases called codons will be translated, they'll be read, they'll be decoded in order to bring in amino acids in a particular sequence to build a protein. The genetic code table shows us every possible combination of codon, every single possible combination of 3 of the 4 bases that are found in RNA: A, U, G, and C. Each individual codon codes for a specific amino acid. And that's what the genetic code is. All living things on this planet use the exact same genetic code. Let me say that again because it's very important. All living organisms on this planet use the exact same genetic code. That's another piece of information that we are all related to each other through evolution. Remember, protein, like DNA, can also be described as having multiple levels of structure. So just like we talked about for the DNA molecule, having primary, secondary, tertiary structure, proteins also have primary, secondary, tertiary structure. And of course, proteins have a 4th level called quaternary. Remember, the primary structure of protein is its sequence of amino acids. Every protein has a different primary structure. It has a different sequence of amino acids in it. The secondary structure of protein are the alpha helices and beta pleated sheets that will form when a long linear chain of amino acids starts to fold. There are 20 common amino acids, and those amino acids each have slightly different chemistry. Some of them will be attracted to each other, some of them will be repelled from each other. Those weak attractions will create the folding that occurs for each different protein. The tertiary structure of a protein is that 3-dimensional folded shape. And of course, the quaternary structure is a term used to describe multiple tertiary proteins that come together and function as a unit. Every single protein has its own unique primary structure, its own unique sequence of amino acids. And proteins are big. We're talking about hundreds to thousands of amino acids. Every single individual protein then has its own unique three-dimensional shape, and that shape is achieved through folding. It's that three-dimensional shape that allows each protein to do its own unique job, its own function within the cell. If we change the order of the amino acids in a protein, we will change how that protein is folded, and we will change its function. Now the question is, why would the primary structure of a protein change? Why would the order of amino acids ever change? And the answer is, we could see a change in the code. If you change the amino acid sequence, that means there must have been a change in the codon in the mRNA molecule. There must have been a change in the sequence of bases in that mRNA molecule. And in order for there to have been a change in the mRNA molecule, there must have been a change in the sequence of bases in the DNA molecule. That is what a mutation is, a permanent change in the sequence of bases in the DNA molecule. If you change the order of the bases, the sequence of the bases in the DNA molecule, and the cell does not catch that change, the cell does not recognize it and repair it, it will become permanent in the DNA, and it will then be a mutation. Any mutation in DNA will cause a change in the order of the bases in the RNA that is produced from the DNA. And when you change the order of the bases in the RNA, you have changed the codons in the RNA, and therefore you have introduced the possibility of changing the sequence of the amino acids that are produced from that RNA. What causes mutations? Well, a couple of different things. Most people that you talk to about mutated DNA know that there are certain chemicals that can do it, what we often call carcinogens or mutagens. Certain forms of radiation can do it. Certain viruses can do it. Long-term, significant chronic inflammation in living cells can lead to DNA mutations. These are the kinds of things that most people will think about when they think about mutated DNA. But we must remember, too, that DNA gets mutated routinely when DNA replication occurs. DNA replication involves an enzyme, and enzymes make mistakes. They are not perfect. They make mistakes. When the DNA polymerase enzyme performs DNA replication, in other words, there are times when it will put the wrong base in the new DNA molecule. Now, inside eukaryotic cells, there are multiple repair mechanisms. There are proofreading mechanisms and repair mechanisms that will correct most of those mistakes. But some of them will persist. Prokaryotic organisms also have repair mechanisms, proofreading and repair mechanisms, but not as extensive mechanisms as eukaryotes have. When a mistake persists, when the systems fail and a mistake persists, you've now got a change in the sequence of bases, the primary structure. That means sometimes a change in gene, a change in a gene. And that could possibly create a change in the protein that gets made from that gene. Mutations that are not caught by the cell and repaired are going to become permanent. And when they're permanent, they get passed on during cell division to new cells. Mutations are permanent changes in the sequence of bases in DNA. Therefore, mutations are permanent changes in the genotype, the sequence of, or the collection of alleles. At least theoretically, at least possibly. It is possible for mutations to occur outside of the DNA that composes genes. It is possible for mutations to occur in other types of DNA that does not code for protein. But if mutations occur in genes, now you've had a change in the genotype. Mutations in DNA can cause changes in codons, because if you change the order of the bases in the DNA, you've changed the order of the bases in the RNA, and that leads to a change in the codon. Now, here's where it can be a little confusing. If a mutation changes a codon enough, that codon will now code for a different amino acid. If you change the order of the amino acids in that polypeptide that gets built at the ribosome, you have produced a new protein. And if you produce a new protein, you have changed the organism's phenotype. Because remember, it's protein that creates traits in a cell. And it's the collection of traits that is the cell's phenotype. Not all mutations lead to a different protein. That's probably the hardest thing for some students to grasp when we talk about mutations. It is possible to change the genotype of an organism without changing its phenotype. Remember, mutations in the third position, the third base of a codon, are usually silent. The genetic code that all living cells use is redundant. Now, what do we mean by that? We mean that for each amino acid, there is more than one codon that codes for it. In fact, for many amino acids, there are 4 codons. And those 4 codons differ in the third position. Look at serine. If the ribosome comes upon a codon with the sequence UCU, remember that's in the RNA, the messenger RNA. If it comes upon a codon with the sequence UCU, it will decode that as serine, and a serine amino acid will be placed at that location in the polypeptide. If the codon were mutated to read UCC instead, the cell would still place a serine in that location. In fact, you can put any base in the third position and the cell will still place a serine there. This is what we mean when we say there's a little bit of wobble in the genetic code. Mutations in the third position don't cause any problems. They don't change the protein at all because the same amino acid gets placed most of the time. There are a couple of exceptions. If you look through the genetic code table, you'll see that there are a couple of amino acids that are only coded for by 2 codons. For example, cysteine and tyrosine. So there's a little wobble in here. You can put a U or a C in the third position and still get a cysteine placed. But if you were to put an A in there or a G in there, you'd have a different effect. Most amino acids are coded for by 4 codons, and the 4 codons differ in their third base. Any base can be in the third position, and you'll get the same amino acid placed at that location. There are some codons, though, there are some amino acids that are coded for by only 2 codons. So it's a little more likely in that case that you'll have a change in the order of the amino acids if that area of the mRNA and that area of the DNA are mutated.